like to thank the organizers for inviting me to talk in this audience. This is a very different audience than that of the geneticists that I usually speak to. So it's really, I think, will allow me to highlight, mostly towards the end of the talk, how I think it'll be important to integrate across some of the really beautiful and detailed studies that have been described over the last day and a half with the kind of work I'm doing. So. The driving question, really, for me, is not so dissimilar to, I think, the driving question for many of you, except I want to know how the circuitry of the brain is abnormal in the diseases that I think influence, impinge on cognition, impinge on emotion, impinge on perception. If I translate that, those diseases would be schizophrenia and bipolar disorder, and that's largely what my lab has been studying now for about the past 15 years. And the way I've chosen to go about this is to use molecular genetics or genomics to try to identify anchors that we can use to better understand the abnormal circuitry that's at play in these diseases. And the way I've been doing this over the years, and I think this is only recently begun to pay off, is to try to fully characterize the entire genomic architecture, all of those things that lead to increased risk for these diseases. So the current theory of what is wrong in schizophrenia and bipolar disorder and autism, and this talk is way too short for me to be able to cover autism at all, is that they're neurodevelopmental abnormalities. This is just a diagram that will be familiar, sort of, to all, much more familiar to all of you in the room because you study various aspects of this. But of course, you know, the proliferation and migration of neurons is largely occurring before we're born, and that there's a great increase in arborization and myelination, and then over time there's pruning of excitatory synapses and maturation of inhibitory synapses. And of course this leads to a theory in particular for schizophrenia, although it's really becoming much more appreciated the extent to which cognitive deficits play a role in bipolar disorder these days, that there's potentially, because the prodromal stages of these illnesses and the onset of first symptoms correspond with some of the maximal phases in which the pruning is going on and the maturation of myelination, that abnormalities in these processes may in fact be involved in schizophrenia. These follow older theories that dopamine system was involved, serotonin system was involved, glutamate system was involved. None of these theories yet have explanatory power for a particular person or have led to any kind of mechanistic understandings. We don't know yet whether that's because the phenotype is not measured well, whether there's a great deal of heterogeneity in what's causing the diseases, whether there's so much redundancy in the circuits. And so these are the kinds of questions that remain open. But what is crystal clear is that these are substantially genetic diseases, the largest risk factor for developing schizophrenia and bipolar disorder. And so, of course, schizophrenia is the classic, is the disease that's classically presents with cognitive abnormalities and delusions and hallucinations. Bipolar disorder classically presents with mania, abnormal mood, which may or may not alternate with depression. But in the general population, the risk for these is substantial. There's lots of morbidity associated and mortality associated with these diseases and an enormous global health burden. But what's important to note is that the risk to first degree relatives for both of them is something on the order of tenfold. We know this from 100 years of studying large families, monozygotic concordance rates, so the chance that if one twin has schizophrenia, his or her co-twin will have schizophrenia is markedly elevated when the twins are genetically identical as opposed to when they're not. And this leads to heritability estimates, both using older criteria and newer criteria that are relatively consistent with 80 or 90% of the individual liability to these disorders lying in the genetics. Now, all of that being said, up until about 2007, if you asked me what the total genetic architecture was, what among all the kind of different abnormalities that have been observed in the DNA in a variety of different diseases, other diseases were playing a role, I'd have to tell you that there were none. and that what's happened in the last 4 years, so I'm going to detail some of this even though it's not as much an open question area, is that the quality and the quantity of the information that's available about what the genetic risk factors for schizophrenia and bipolar are, and also for autism, have substantially changed. And with, excuse me, with a characterization of copy number variation, several forms of common variation, and more recently, as next generation sequencing technologies are coming online, with rarer and rare SNP variation. So the first thing I have to tell you is that there were 20 or 30 years of traditional genetic studies. So this is the kind of study that one would use to find the Huntington's disease gene, and it's been spectacularly successful for 1,500 or 2,000 or so Mendelian or single gene disorders. So that is, you look at a large series of pedigrees and you ask questions about what part of the genome segregates. And what you find is that in schizophrenia, and this is true, although it has not quite been done in this spectacularly large a sample, that there is no single gene or no single handful of genes that have mutations in them that account for a substantial number of the cases of patients who get schizophrenia. So 32 studies have been done, over 3,000 pedigrees, 7,000 affecteds have been looked at. This data's all been put together. And what you can see, this line just represents the line that you need to achieve to have found a result that was not by chance, having tested all these, the number of markers that are tested across the genome. Nothing crosses that line. And so what we can say now is that there is nothing that's giving you a 50-fold or 100-fold increased risk or a 1,000-fold increased risk of developing this disease in any one single gene. And in fact, this is such a large study that it had the power to detect really small effect sizes. So even if there were a single gene or a small number of genes that increased risk just a little bit, we would have been able to detect it in this study. So something else must be going on. And so the first clue to what that was came from a series of studies in 2008 that my lab and several others participated in, where we were able to isolate and identify that there were very specific parts of the genome that were missing in patients with schizophrenia. These were, in general, very large, and that if you carried one of these loci, you had something on the order of a 10 to 30-fold increased risk of developing the disease. This is the largest genetic risk for development of schizophrenia that exists, except for having a monozygotic twin who has schizophrenia. What we and others were also able to do was because these studies were so large, we were able to show that there was, in addition, a subtle excess across the genome of the rate of rare deletions, and that this was most elevated in those that only ever occurred once, the rarest cases in our data set, implying that maybe the genes that were interrupted or intersected with that set were those that might really be leading to disease. The next big surprise, though, was that these very same DNA lesions can produce very different diseases. And this has become a theme both for all the forms of variation that we've been finding in association with schizophrenia and bipolar disorder. So you can see here that there's a whole variety now of deletions that are being followed up and studied, and that they cause a whole variety of diseases. And in fact, more and more, it's becoming clear that duplications, which weren't initially assumed to be pathogenic because they're much more common in the genome than deletions, are now being reproducibly and statistically compellingly associated with diseases. So where the specificity occurs here is not at all apparent yet. Whether this is a function of experience or genetic background or other epigenetic factors remains to be unknown. People carrying these loci are relatively rare, so it's a very active area of investigation for geneticists. So de novo CNVs, those CNVs that were not inherited from parents, represent a very special case of a special case for trying to understand what pathways might be interrupted or might be part of the pathology of the disease. And so we undertook a really the largest study that's been done looking at the inheritance of copy number variation in children from their parents. And so here we hypothesized that these things should be the CNVs that are very large should be rare due to negative selection, and that would be, they would likely recur, the fact that they recur in the same places over and over again would be occurring through de novo mutation. With these observations, of course, consistent with the well-known reduction in fecundity, about half what is found in normal individuals in patients with schizophrenia. And so we wondered whether they're enriched in a particular biological hypothesis. We did this in over 600 parent and child trios. And because there's a lot of testing involved, we decided to just test one specific hypothesis related to what is often called neurodevelopmental genes. There had been some preliminary work in the literature suggesting that these might be relevant. Of course, the cognitive symptoms are prominent in schizophrenia. And so we chose to direct molecularly to investigate glutamate receptor complexes at the postsynaptic density. And here, what's different is these are not literature PSD, but these were directly PSDs that were directly precipitated by Seth Grant and analyzed by LC mass spec. And so we've directly looked at the proteins in a variety of complexes, and what we found was that they were in fact enriched in the postsynaptic density. And what I've shown you here are the number of de novo CNVs that arose in genes that are in this set of 664 proteins that came down with the postsynaptic density out of the 34 de novos in our set. This is a control set of 1,300 individuals from Iceland. We've done this with a number of controls because there are all kinds of statistical issues related to bias, and I feel very confident that these results do not result from any kind of systematic bias here. And so you can see that in general, the proteins in the ARC complex or the AMPA receptor complex or the NMDA receptor complex are largely contributing to what we're seeing in the postsynaptic density. I haven't put on this slide the number of other pathways that were also investigated, synaptic vesicles, things of that nature, that all of which were negative. In terms of common variation, there's been substantial success also recently in identifying risk factors by coming together as a large group. About 3 or 4 years ago, the Psychiatric GWAS Consortium got together, understanding that in order to make progress in the field, it would be absolutely critical to come to put the large data sets that were being, at that point, investigated and collected together. And so for schizophrenia recently, I'm sorry, the screen here is looking funny. So for schizophrenia recently, we've just been able to analyze almost 10,000 schizophrenics and a large number of controls. All of this data really required an enormous amount of sharing. We picked a neutral country, which was Holland. There's a server there that all of the data was uploaded on, and all of the raw data was in fact analyzed on that server so that everyone agreed not to pull down the data. But I think that it really shows that there is the possibility for this kind of large-scale effort. And what we now have are 5 new loci. We were able to confirm 2 prior loci. 2 didn't make it through into our replication sample but have been independently replicated. So now we have about 10 new loci. For bipolar disorder, there were 2 results. One that had been sort of nudging its way up, the L-type voltage-gated calcium channel, and ODZ4, which is related to a Drosophila pararule gene. For all of these findings, there's consistency across. They're mostly all common variants. The principle is the same as for any other disease that's with complex genetics that's been looked at. That is that the effect sizes for each of these individually is relatively small. The important thing, I think, to notice is that in both of these cases, the replication samples indicate that there's a lot more to be found. So of those SNPs that were attempted in replication, they overwhelmingly had nominal p-values in the same direction of effect, which we wouldn't have expected by chance. And so we feel that we're probably on the same sort of trajectory that other disorders like adult height and Crohn's disease are on, which is that as you begin to increase the sample size, you're able to go into a substantial depth and begin to unravel some of the pathways that are involved. So the most associated region is the major histocompatibility complex. This is an extraordinarily complicated area with strong LD that contains both MHC class 1 region genes, which have been implicated in brain function, as well as class 2 region, factor receptor gene cluster, histone gene clusters, as well as a lot of brain genes. This will be a very difficult area to follow up. We've generated a totally novel hypothesis here with this observation that the best signal that we now have, which is an extraordinarily large signal in a psychiatric genetic study, actually is leads us to believe that a microRNA, which is miR-137, is involved in the pathology. Additional evidence of this is that when we take target scan and look at the predictions of microRNA miR-137 targets, we find that 17 out of the 301 are predicted. 17 of the 301 predicted targets are very strong association hits with schizophrenia. Independently. In fact, the top 3 were 2 of the top 4 were genome-wide significant on their own. So let me skip then. Similarly, calcium signaling is another novel hypothesis that's emerged directly from the genetic data that has a lot of support from data that existed in the field prior to this. So we've known for a long time that calcium channel blockers have been used with variable success to treat bipolar disorder and migraine, of course, which has individual point mutations in some of the calcium channel genes, are overrepresented among bipolar patients. There is a rare Mendelian form called Timothy's syndrome with multiple organ system abnormalities that has been with familial features that resemble autism. And now we know from these studies that the calcium channel subunits as a group are enriched in bipolar disorder, and in fact they're also independently associated with both schizophrenia and major depression. So understanding the role of calcium channels, I think, will be critical in sort of reinvigorating that, which has not been seen as a particularly sexy field for follow-up either by the drug companies or by neurobiologists. I think it needs to change. The genes that we are identifying in these groups of association signals are not a random set. And so what I've done recently is looked both at bipolar disorder, and this was recently published for schizophrenia, and asked the question, if we use a set of data that have been derived from the Harvard Brain Bank, where we've looked at expression QTL, so SNPs that control the expression level of proteins, and ask a question, is there enrichment of those among our association signals? It's strong both for bipolar disorder and for schizophrenia. And this implies that the set of enriched associations are not at all random with respect to the association data. A couple of years ago, we also we asked a question that was considered heretical at the time, which was, are there many in even thousands of genetic contributions that might be too small to measure individually, but we might be able to measure in aggregate? And the answer to that question was yes. And what we were able to do was to take a large discovery sample and ask questions about the top 1,000, 2,000, 30,000, 50,000 association signals and build a predictor, and in a target sample show that they were very statistically compellingly associated with the same difference between schizophrenia and bipolar disorder. The big surprise, of course, was that, the control was, of course, that predictor had no ability to predict in all of these random non-psychiatric diseases, but the predictor did predict very well the difference between bipolar patients and controls, providing some of the very first strong molecular genetic evidence that lots of the alleles might be shared. This has now been enlarged and done in the reverse direction, so bipolar disorder, target allele, bipolar alleles, that bipolar disorder alleles predict bipolar disorder as well as schizophrenia, so the inverse experiment. What the lab is spending a great deal of time working on now are the research questions that will occupy geneticists in psychiatric genomics for the next few years, which have to do with do known schizophrenia CNVs harbor rare point mutations? Are there genes where multiple risk alleles of moderate effects occur? And can we do some statistical and technical development that will allow us to make use of exome sequencing? We have a wonderful collaboration with Christina Holtman and Patrick Sullivan, in which we have a great deal of information at both epidemiological and otherwise on 35,000 probands with schizophrenia, of which we've now collected 5,000 cases and 7,500 controls. We've been obtaining both common variant information, so they've been GWASed, and now we've completed the first couple of waves of exome sequencing as well as a targeted study. And so let me just tell you the results of the targeted study. The hypothesis, of course, is asking a question about whether common copy number, the large copy number variation, the big deletions that we've found are pointing to genes that individually increase risk of schizophrenia. That might be a theory. Also, the singleton genes might point us to genes that would have other genetic abnormalities within them. And so we have pooled about 250 of those genes and resequenced them in 750 cases in controls, and then asked questions about, on balance, all of the amino acid changing SNPs that were found in those genes and whether they were associated as a group with increased risk of schizophrenia. And in broad terms, what we were unfortunately unable to find was any evidence that there were single genes, either for the singletons, that showed a pileup of mutations or amino acid changes. So what we're not able to provide is any support for the hypothesis that the CNVs per se are pointing to a small number of hotspots, or the large CNVs in particular, pointing to a small number of hotspots within them. And this is probably not surprising at all, given the fact that as the technology for looking at smaller and smaller CNVs has evolved, we have not picked up more CNVs that are causing schizophrenia, even though the number of CNVs that are detected are much larger, probably suggesting that you really need to knock out a substantial number of genes simultaneously to produce these kind of complex brain phenotypes. Similarly, the basic statistics and things on our first set of 500 cases and 500 controls were excellent. I won't go, I obviously put too much of these background slides in, but the sequencing itself needs a great deal of care and QC, and everything about it thus far looks very good. Our initial scratching the surface at what we've been looking at are really just asking this question again, just like I described for the targeted sequences, is are there a burden of rare variants that are found in cases versus controls? For example, are there a whole bunch of stop codons in a particular gene in the cases and not in the controls? And the answer to all of these questions are largely there is no smoking gun here. So there are some 5 to 0s in the cases and the controls, but there are equally as many if you look in the opposite direction. And so where we are right now is that there is no really strong single genic locus in our initial look at the data, even if we condition on polyphen functionality or any of the sort of traditional things that are done in the literature. So we have a huge amount of ongoing work, and this is a big piece then of what needs to be done in genetics. We need to extend our samples both to be able to look and bite off more of what the common variation is so that we can begin modeling together common variation, CNV variation, rare variation. And so we have a great deal of work in all of these areas going on in the group. But the take-home message here really is that the genetic architecture has changed substantially in the last 4 years, and we can now fill in some of the contributions. All of these contributions will likely increase over the next few years as we collect additional data. But the question is, how do we then go and integrate this information so that we can do something that will ultimately lead to an improved mechanistic understanding of what the circuits are that are awry, that will allow us to predict for individual patients whether they might develop the disease or not, and will actually have therapeutic potential. And so Eric Schadt talked about some of this yesterday, so I don't have to go through much of the preliminaries. But one of the tacks we're taking is assuming that the data is in fact quite complicated and that we're going to need to understand and that the circuits that are involved are complex and that we will need to feed in together in complicated ways as much data as we possibly can on the normal brain, the circuits of the normal brain, the networks of the normal brain, in sort of gene language, also looked at in terms of what we understand about the differences between schizophrenics and controls in brain imaging. And so here he showed yesterday that we can develop the same sort of strategies for expression microarrays using structural MRI and functional MRI. We can cluster the data. The clusters show that there are differences between cases and controls that are, at least initially in this first very preliminary data set, statistically significant. So the role then is, and the path forward in the future, I think, is to construct a whole variety of gene expression and imaging networks that are then tempered by an understanding and an increasingly evolved understanding of what the genetic underpinnings are. Here, we can then, of course, feed in additional information, either as in the form of Bayesian priors or in the form of covariates, so that we can begin this process of understanding what the networks are. And my particular interest is in understanding what the key drivers are, because what I'd like to be able to do is to do the kinds of things that are more often done in this audience, which is to have in a dish some cell type that I can manipulate in a well-defined way. And so here we're also building with my old colleagues at the Broad and with my new colleagues at Mount Sinai, fairly robust banks of fibroblasts and induced neurons and induced pluripotent stem cells, so that we can begin the process on large scale of studying the role of those genetic factors in their normal genetic human background and perturbations thereof. So that's the end of my talk. What I wanted to say is that I do think that the open question for me is how in the setting here do we connect the wealth of information that was talked about over the last few days that's derived from lots of different systems, lots of different circuitry, particularly mice, to advance the understanding of human brain diseases. There are lots of people that participate in this. I have a long-term collaboration with Sean Purcell, and all of the genetic data that I talked about is work that we've done together. And then finally, most of you still remember that me as a person from Harvard and the Broad, I've only recently, about 6 months ago, moved. Here's my lab now at Mount Sinai. And there are lots of faculty and postdoc openings as we build a new Center for Scientific Medicine, and in particular, individuals of computational background who are interested in moving forward with either the specific or the network-based kinds of studies that I described are welcome to contact me. So, sorry that I went a little long.